Darwin's concept of gradual changes and survival of the fittest is still functioning. But in addition, we have this other genetically driven, punctuating, sudden change capacity that is really characteristic of the HOX genes. HOX genes are the blueprint for all living creatures. HOX genes were present at the beginning of your life. All mammals, the class of organisms to which we all belong, begin life following the union of an egg and sperm, resulting in a single-cell zygote. The zygote immediately begins to divide, producing a ball of undifferentiated cells. Soon, the spherical cluster of cells takes on the shape of an embryo as cell differentiation forms specific tissues such as skin, muscle, neurons, fat, and organs, and homeotic genes organize the body plan, starting with the head. This process is known as morphogenesis. Morphogenesis is controlled and directed by a special set of genes known as the homeotic genetic system. The term homeosis refers to anatomical replacement of one body part by another. The homeotic system is composed of some 200 genes, including a subgroup of homeobox genes named HOX genes for short, consisting of a group of 39 HOX genes located in all vertebrates. In 1984, a HOX gene mutation resulted in a misplaced entire leg appendage, normally located on the thoracic segment, to have grown on the fruit fly's head, replacing an antenna. This has become known as the famous antennapedia mutant. It is this unique phenotypic function of HOX gene mutations, known as homeotic transformations, that alerted geneticists to the unique functionality of HOX genes. The left upper image is a normal fruit fly's head. In the right upper image, a leg appendage is growing at the normal location of its right antenna. In the lower pictures, on the left, there is a whole normal fruit fly, whereas on the right, a bithoracic mutant is shown with 2 complete thoracic segments and 2 pairs of wings. There are 2 major HOX gene complexes in fruit flies known as antennapedia and bithoracic complexes, consisting of a total of 8 HOX genes as listed with abbreviations. The function being performed in this image of RNA polymerase II, located in the cell nucleus of all eukaryotes, is known as transcription and represents what is considered the heart of molecular biology. RNA polymerase II is shown in purple in molecular detail, meaning that each tiny ball represents an atom. The double-stranded DNA is held in the jaws of the RNA polymerase, in which the 2 strands, the bright orange and the yellow orange, have been separated or unzipped. The bright orange is actively being transcribed. It is being copied onto a single strand of messenger RNA colored green. The process of transcription involving copying, distributing, and interpreting genetic information has become known as the central dogma of molecular biology. Information management is at the heart of all of life's activities. Homeobox genes display a distinctive structure and configuration, identifying them as a unique set of genes. A unique feature of each homeodomain sequence is a region containing 3 precisely configured alpha helixes, enabling direct contact with specific regions of the DNA molecule. In the illustration, the 3 alpha helixes are represented by coiled ribbons differentiated by color and size. The largest red ribbon motif represents alpha helix number 3, that is in contact with the major groove of the DNA molecule, and the N-terminus arm in contact with the minor groove, completing a lock and key molecular interaction. Number 2 is represented by the shortest spiral ribbon colored yellowish green. Number 1 is represented by the greenish blue intermediate sized alpha helix. Helixes 1 and 2 are antiparallel, and both are perpendicular to number 3. They are tightly packed together by hydrophobic interactions. In this manner, the homeotic system turns a gene on or off in the correct order and at the correct time, resulting in the transcription of genetic information essential to the living organism or growing embryo. The acquisition, storage, distribution, and dissemination of accurate information is absolutely essential in all living organisms. Spatial collinearity is a unique feature of homeotic genes. Generally, non-HOX genes appear scattered throughout chromosomes with no conceptual pattern or predictable location. In contrast, HOX genes are like, open quote, islands of compatible, comprehensible structure, close quote. Spatial collinearity refers to the organized location of the HOX gene clusters on a given chromosome. The location of the gene on the chromosome is essential to its normal function. Homeobox genes provide regional identification of the normal locations of body structures. They provide the information needed by new cells, enabling them to undergo differentiation to produce the specific features and structures at the right time in the correct locations along the anterior-posterior axis of the developing embryo. For example, in insects, each HOX gene is responsible for a different segment of the body, such as the head, thorax, or abdomen. This schematic drawing illustrates spatial collinearity. In A, the color coding indicates that the location and relative positions of the 8 HOX genes on the chromosome of the fruit fly corresponds to the region of control on the specific segment in the larvae as well as in the corresponding segment of the adult. The timing of their action also sequentially controls the developmental progress from the head toward the tail, resulting in a HOX gene controlling the entire body plan anatomically and sequentially. In B, the broad curved arrows indicate functioning cis regulatory molecules that can influence dynamics of gene expression. C and D illustrate specific and extensive phenotypic alterations that may result from cis regulatory molecular action resulting from deletion mutations. Extreme examples indicate that they can transform a fruit fly into a dragonfly-like or a spider-like insect. E illustrates that both deleted or added whole abdominal segments can result from cis regulatory molecular action. This image illustrates the remarkable degree to which HOX genes are conserved across multiple evolutionary levels. On the left, there are samples of different organisms along the evolutionary tree of life, starting with C. elegans, which is a primitive roundworm approximately 1 millimeter in length. It is a member of phylum Nematoda. It is bisexual, lives primarily in soil, is non-segmental, and has been extremely scientifically analyzed. It has been described as, open quote, about as primitive an organism that exists, which nonetheless shares many of the essential biological characteristics that are central problems to human biology. The next organism in the evolutionary lineup is the fruit fly, which is an outstanding model for scientific investigation, particularly homeotic genetic studies. It has 4 chromosomes, possesses 8 HOX genes, all located on 1 chromosome, has a short life cycle, striking genetically controlled eye colors, can rapidly reproduce large numbers of offspring, and can be maintained economically in a laboratory. It is the most useful model for studying genetically transmitted human diseases, since approximately 75% of homeotic genes that induce human diseases are shared by fruit flies. Fruit flies were the primary organism studied by Gregor Mendel, 1822 to 1884, who is among the greatest geneticists of all time. The third organism is the Florida lancelet, a representative of the vertebrate subphylum Cephalochordata. Cephalochordata display the same basic plan as vertebrates, in which structural integrity is maintained by a notochord that extends the entire length of their body, occupying the anatomical location of vertebral bodies which subsequently develop in vertebrates. They are histologically and anatomically much simpler than vertebrates. They have 14 HOX genes with no duplications. The fourth evolutionary stage is represented by 2 mammalian vertebrates, common house mouse and Homo sapiens, which share the 4 identical HOX gene clusters. The vertical red and green lines indicate experimentally documented functional equivalence between genes in the different clusters on the different chromosomes. This documents significant functional redundancy in the homeotic gene system, providing significant conservation stability. The term conserved refers to both their widespread distribution throughout all living organisms and their history of having been accurately copied from generation to generation since the near beginning, if not at the beginning, of evolutionary life. They remain essentially unchanged in chemical composition and function since the earliest forms of evolutionary life more than 500 million years ago. The level of conservation is unique and unsurpassed by any other group of genes. In 1969, Robert Whittaker proposed a five-kingdom system for the classification of all living organisms that has become generally accepted. The homeotic system exercises master control throughout the kingdoms of plants, animals, fungi, and numerous single-celled eukaryotes in Protista kingdom, representing four of the five kingdoms of living organisms. Duplication and divergence. Duplication and divergence evolves new genetic loci and new metabolic pathways. There have been two nearly complete HOX gene duplications having occurred at or near the origin of evolutionary planetary life, resulting in the four clusters now present in all vertebrates. The evolutionary process achieved by the beneficiaries of the gene duplications and vertebrates, inclusive of mammals, have far outstripped the invertebrates. In 1970, Susumu Ono proposed that new genetic loci were essential for progressive evolution. Another well-documented HOX gene duplication occurred in ancestors of the spiders and scorpions during the late Cambrian era, resulted in sudden appearance of book lungs in air-breathing scorpions, consistent with Ono's proposed hypothesis. Punctuated equilibrium. The unique characteristics of HOX genes in which they are capable of bringing about extensive reorganizations of body plans and at the same time do so rapidly because of their control of large numbers of other genes, combine to explain and support the theory of punctuated equilibrium that has gained significant attention and acceptance as an advancing explanation of why there are so many gaps in the fossil records. The Urantia book description of a crucial stage during the final step in human evolutionary development describes the theory of punctuated equilibrium in action as follows, open quote, a little over 1 million years ago, the dawn mammals suddenly appeared. These were active little creatures, about 3 feet tall and could easily stand erect. They had a primitive opposable thumb, as well as a highly useful grasping great toe. From this point onward, the pre-human species successively developed the opposable thumb while they progressively lost the grasping power of the great toe. These aggressive little animals spread over the Mesopotamian peninsula for more than 1,000 years, constantly improving in physical type and general intelligence. This new tribe had taken origin from the highest type of lemur ancestor. The next epoch-making development occurred the sudden differentiation in the ancestors of the next vital step in the evolution of human beings. We will now examine a series of major, suddenly occurring evolutionary developments characteristic of punctuated equilibrium, in which HOX genes controlled the evolutionary process. The Urantia book states, suddenly and without gradation ancestry, the first multicellular animals make their appearance. The trilobites have evolved, and for ages they dominate the seas. From the standpoint of marine life, this is the trilobite age. The appearance of trilobites in the fossil records of the Cambrian explosion 521 million years ago is among the most remarkable success stories of our entire biological evolutionary history. Trilobites were particularly suited to fossilization, having a calcified exoskeleton and existing primarily as an aquatic organism combined to make an immense fossil contribution. They existed for 270 million years. Over 22,000 species have been described. They reached a large size, measuring more than 70 centimeters and probably weighed as much as 4.5 kilograms. A most reasonable terminology to characterize the biological progress that happened during the Cambrian era is the Cambrian explosion. And the geological evidence indicates that the homeotic genetic system was a major contributing factor. Paleontologists recognize the importance that HOX genes have played in evolutionary development of the many organisms of the past. A phylogenetic analysis used to recognize homeotic evolution in trilobites reached the conclusion that the homeotic evolution commonly occurred among Cambrian trilobites. Trilobite bowel plan contains 8 body regions or segments. The cephalon contains the frontal lobe, glabellar, and occipital ring regions. The thorax contains the anterior thoracic and posterior thoracic segments. The pygidium contains the articulating ring, axial and terminal piece regions. Open quote, trilobites being amphipods probably contain 8 major HOX genes that controlled their segmental types. The Urantia book states the larger mollusk or cephalopods grew to 15 feet long and 1 foot in diameter. This species of animal appeared suddenly and assumed dominance of sea life. Following the disappearance of the trilobites, the oceans were rapidly taken over by the larger mollusk and cephalopods. They grew up to 15 feet in length and soon dominated the seas. They consisted of a large, diverse group of marine animals, including octopus, squid, bivalves, snails, clams, oysters, conches, sea slugs, and periwinkles. Studies of homeotic genes in mollusks, open quote, revealed an overall number of 11 HOX genes in 7 out of 8 molluscan class level taxa, close quote. Homeotic gene studies performed on one of 3 prominent bivalve subclass members of the phylum Mollusca, Acrinita, reveals extensive transcribing activity throughout the larval stage. The active transcription process is documented by the yellow fluorescence throughout major portions of the larval embryo, which is displayed in orthogonal sections. This is a common method of tracing HOX gene activity as it is directing the body plan, segmentation, timing, and location of gene activity along the anterior-posterior axis of the embryo. Phylum Mollusca is widely recognized as one of the most diverse organismal groups, having populated the Adiacaran oceans around 550 million years ago. Once again, we observe the developmental phenomenon of amazingly rapid evolutionary progress in which the impetus was the work of an ancient, highly conserved, homeotic genetic system. The Urantia book states, toward the close of the final Silurian submergence, there is a great increase in the echinoderms, the stone lilies, as is evidenced by the crinoid limestone deposits. Soon thereafter, and suddenly, the true scorpions, actual air breathers, make their appearance. HOX genes are considered the major genetic impetus to the development of air-breathing scorpions that suddenly appeared following domination of the seas by mollusks and cephalopods. Relatively recently, discovery of an ancient whole genome duplication, WGD, event occurred in an ancestor of spiders and scorpions. As a result, two HOX clusters appear to have been fully retained in families and clusters in arachnopulmonates. Overall, our results show a high level of retention of homeobox genes in spiders and scorpions post-WGD, which is likely to have made a major contribution to their developmental evolution and diversification through pervasive subfunctionalization and neofunctionalization and paralleling the outcomes of WGD invertebrates. Scorpions represent a transitional form bridging the gap between marine and land-based animals. This required development of respiratory organs known as book lungs. In scorpions, this consists of 4 pairs of plate-like or sheets of highly vascular tissue arranged like pages in a book. These are exposed to the exterior, providing a large surface area for oxygen and carbon dioxide exchange. Book lungs have been studied in an exceptionally well-preserved fossil arachnid, Trigonotarbeta, from the early Devonian era, located in Scotland, dated approximately 410 million years ago during the late Cambrian explosion, using non-destructive imaging methods, identified fossil trabeculae for the first time, establishing a permanent airspace for respiration. Open quote, thus showing unequivocally that trigotarbids were fully terrestrial and that the microanatomy of the earliest known lungs is indistinguishable from that in modern arachnida, end quote. The Urantia book states, the earth was being overrun by new orders of land vegetation. Heretofore, few plants grew on the land except about the water's edge. Now and suddenly, the prolific fern family appeared and quickly spread over the face of the rapidly rising land in all parts of the world. Due to a genetic duplication and associated disruptions of the fern genome that occurred approximately 60 million years ago, we are unable to reconstruct the role of HOX genes in the rapid spreading of ferns. The Urantia book states, when the seas were at their height, a new evolutionary development suddenly occurred. Abruptly, the first of the land animals appeared. These air-breathing amphibians developed from the arthropods whose swim bladders had evolved into lungs. Suddenly, large numbers of amphibians permanently abandoned their marine existence. The plants and grasses were spreading rapidly over the land, providing food for the animal invasion taking place at this time. The homeotic genetic system was well established, resulting in rapid growth and development, mobilization of animals in general, and rapid expansion of vertebrates everywhere. The first force began developing. Plant life was luxurious, providing nourishment for the newly arrived land animals. Many of these amphibians had already evolved lungs from swim bladders. Climatically, this was a relatively warm period. The level of atmospheric carbon dioxide was dropping rapidly as the oxygen level was increasing, resulting from widespread photosynthesis by the extensive plants, particularly the ferns. These atmospheric changes greatly benefited the rapidly expanding terrestrial invasion by land animals. The Urantia book states, 140 million years ago, suddenly, and with only the hint of the two reptilian ancestors that developed in Africa during the preceding epoch, the reptiles appeared in full-fledged form. They developed rapidly, soon yielding crocodiles, scaled reptiles, etc. During the Triassic period, reptiles were recovering from the Permian-Triassic mass extinction, thought to have resulted from dramatic climatic changes from an arid period to an extremely wet climate known as the Carnian pluvial episode, dated 234 to 232 million years ago, consisting of perhaps 1 million years of constant rain, after which, once again, it became arid. Other examples of rapid variations in reptilian population occurred during the Jurassic period, known as the Mesozoic, which followed the Triassic period, extending from 201.3 million years to 145 million years ago, extending for more than 56 million years. This is generally known as the Age of Dinosaurs. During this period, the largest land animals to have ever lived on Earth roamed the western states, hatched from eggs, fed largely on foliage, grew to enormous sizes, died, and were then encased in the sand and mud of the Morrison Formation and subsequently fossilized. One of the most concentrated displays is located at Dinosaur Monument in Utah, in which a cluster of fossilized dinosaurs are displayed on a hillside now housed in a building providing a spectacular public display. The Urantia book states 90 million years ago, the angiosperms emerged from these early crustacean seas and soon overran the continents. These land plants suddenly appeared along with fig trees, magnolias, and tulip trees. Angiosperms are known to be the most highly developed and most advanced plants on Earth. They are by far the largest groups of plants, making up 70 to 80% of the various species. They are generally referred to as flowering and seed plants. More than 300,000 species of angiosperms have been identified and are classified into 8,000 genera. They live in all regions of the world. They're capable of living in all climatic zones and have adapted to a wide range of habitats. To date, approximately 5% have undergone analysis of their genome size. Genetically based on their homeobox genes, they are classified into 14 classes, corresponding to 14 classes of land plants. It is difficult to overemphasize the importance of the sudden explosive arrival of angiosperms. They quickly became the major food source for the rapidly expanding animal population. Seeds based on a sexual reproduction in which the male and female gametes are combined to produce a diploid organism greatly expanded adaptability to environmental conditions and climatic changes. In addition, the seeds themselves became a major food source. The sexual means of reproduction is far more efficient and provides far greater variation and competition between plant species than the asexual form of reproduction based on spores relied upon by ferns. The Urantia book states 50 million years ago, the placental type of mammals suddenly appeared. They constituted the most important evolutionary development up to this time. A functioning placenta offers a great advantage in that it provides for relatively mature and well-developed offspring. The presence of mammary glands provides excellent nourishment to the newborn and infant offspring. Superior brain power possessed by mammals resulting from their large brains provide much greater ability to avoid predation as well as adjust to changing environmental conditions and emergencies. Most of our information and understanding of how HOX genes control and contribute to growth and development of mammals is described from our extensive experimentation with mice. Because of their small size, ease of growth, and rapid reproduction, mice have been the experimental mammalian animal of choice for decades of homeotic system experimentation. Since the HOX gene complement in the mammalian genomes are uniform throughout, data derived from the mouse genetic research is equally applicable to all other mammals. M.R. Capecchi at the Department of Human Genetics, University of Utah School of Medicine, has performed extensive studies on the role of HOX genes in mice. He has achieved disruptions of 37 of the 39 HOX genes. He not only described the individual functions of HOX genes during development, but also explored the interaction among them by introducing multiple mutations into the same mouse. From this analysis, he has concluded that in mammals, HOX genes function not only as individual entities, but also as members of a highly integrated network with paralogous genes, adjacent genes in the same linkage group, and even for non-polygous genes. Collinearity in humans. In the human genome, as in all other vertebrates, there are 4 clusters of HOX genes distributed in 4 separate chromosomes. Humans possess more than 200 homeobox genes, of which 39 are HOX genes. HOX genes are a subset of homeobox genes that are directly involved in controlling the body plans of developing embryos, specifically along their anterior-posterior long axis. The 4 clusters represent duplications in which there are slight variations in the number of genes in each family group. Shown are the 4 family members present in each of the 4 loci, along with the direction of individual gene transcription and the directions of the spatial and temporal waves of transcription of the genes in each cluster. Shown on the right is a schematic indicating the relative positions of the human body at which the HOX paralogs are expressed during development. HOX genes control the nervous system. Section A illustrates a standard display of HOX genes that exists throughout the entire vertebrate genome. The 39 HOX genes that populate all vertebrates are displayed with color coding expressive of their unique features of spatial collinearity. In B, HOX genes collinearity is expressed along the anterior-posterior axis of the embryonic nervous system, starting with the hindbrain and extending the length of the spinal cord. In C, the focus is on pontine neurons in the hindbrain, resulting from HOX A5 activity. HOX A5 controls the migration of neurons that occurs during embryonic development of the hindbrain. In D, control of motor neurons by HOX C8 genes are studied. Motor neurons control motor activity such as muscle function. They send signals to muscle cells initiating muscle contraction and other activities such as walking, running, swimming, throwing, and tackling. Conclusions. HOX genes provide the master blueprint for all living organisms. HOX genes can cause sudden advances in evolutionary development, hence many missing links that supports the theory of punctuated equilibrium. They controlled the body plans of living organisms since 550 million years ago. HOX genes have a unique molecular structure known as the homeobox, consisting of 60 amino acid molecules. HOX genes are highly conserved throughout evolutionary history. Our best scientific understanding of the origin of HOX genes is consistent with the Ranchi Book statement. We organized and initiated the original life patterns of this world and planted them in the hospitable waters of the realm. Thank you very much. Very
Ralph,
nice.
thank you. Great talk. We thoroughly enjoyed it. I think we have a number of questions, so we're going to let Jeff start off, and then we'll go from there. Thank you.
So great talk. Couple of questions. You're suggesting that the 39 HOX genes play a predominant role in many or all of the suddenlies.
Hmm.
And do we have any idea what caused those changes? I mean, it's a
Well,
fairly
it's
stable orientation uh, of the gene itself. So what would cause it to be um, disrupted and in such a way as to produce the suddenly?
there are several characteristics of HOX genes which contribute to their ability to bring about the sudden changes that have been observed in biological development, evolutionary development. And one of those is that they actually control large numbers of other genes. They actually are considered master controlling genes, and the best example of that, or one of the most well known, is the fact that a HOX gene mutation can cause an entire extremity, one of the legs on the fruit fly, to replace one of the antennae on the head, which is actually, of course, producing a transfer of a body part from one segment of the insect to a different segment, from the thoracic to the head segment. And that, of course, is the result of a mutant. So it's that phenomenon in which they are able to bring about dramatic changes with no intermediate stages. And that seems to be what happened with this series of sudden changes, sudden advances, sudden dramatic developments, evolutionary developments that occurred, and which is well documented in the fossil records, and and that's the nature of this of these HOX genes. And it's very interesting from my point of view that we have just discovered these a few decades ago. In the 1980s, we first became aware that this was possible. We had never appreciated this potential dramatic uh, evolutionary changes that the genes are capable of causing. Uh, I like to think that this is a reflection of the life carriers who are described as among the most versatile, the sons of God, and they transferred this uh, fertility into the genes that were planted in those uh, inland seas 550 million years ago.
I would like to add one more question, and that is, if are you familiar with the work of Michael Levin, where he controlled the electrical environment that produced an eye in a flatworm in somewhere else in the body, and it grew an eye in a different location. So I'm trying to relate the HOX gene electrical environment with this, body part repositioning. It seems like the DNA is essentially everywhere in the body. So
Mhm.
it's a matter of what genes are expressed in what areas. So the question becomes, how are they expressed, in the case of a gallbladder to become a gallbladder, and a brain to become a brain. And if it's the same DNA, and he's saying that it's more the electrical environment with which they are replicating. And that brings me to my favorite subject, microtubules, because they hold the environment, or they hold the positional uh, orientation of the cell as it's replicating. So, it could be, from my perspective, an electrical environment as managed by the microtubules, which, by the way, are pretty intelligent on their own.
Mm-hmm.
So the next question leading is, once you develop an opposable thumb because of one of these suddenlies, what do you think makes it dominant? Or is it the duplication of it? Is it what makes it stick, so to speak?
Mm-hmm. Well, it appears as a result of our recent understanding of HOX genes, as I mentioned, it's just been within the last several decades. Uh, HOX gene capabilities have been uh, uncovered, have been recognized, which are in addition to the gradual capabilities of genes, which of course was well described by Darwin as the bringing about survival of the fittest. And both of these processes occur in genes. And we have a great deal of evidence that these have both been occurring like intermittently, which has given rise to that recent, fairly new concept of punctuated equilibrium, in which geneticists have recognized that there are these sudden giant steps forward, as it were, in the genetic expression, uh, bringing about completely new body forms and new capabilities, such as the book lung in the scorpion. I think that's one of the most fascinating. Uh, and at the same time, the genes are also capable of the gradual sort of progress that occurred uh, during the 1,000 years on the Mesopotamian Peninsula, where pre-humans were gradually developing a more functional right thumb, left thumb, and at the same time losing their grasping great toe. So this is all part of the genetic uh, functioning that they're capable of doing. And obviously, electrical phenomena are extensively involved in, in nerve transmission and in nerve control and in body functions of all sorts. The contraction of every muscle, each time a muscle fiber contracts, there's an electrical component to that, stimulating, a sudden change in voltage potential across the nerve sheath, uh, and so electricity is very much an integral part of all human activity. Electrical
Indeed,
phenomena, I should say.
indeed.
Ralph, I think and correct me if I'm wrong, but in very recent times, like within the last 20 years, in developmental biology in humans, they of course have been worried, very concerned about fertility, and um, the guess is that way over somewhere between 60 and 90% of when an egg and a sperm actually meet, there's failure.
Mhm,
So really only about
yeah.
10% of those events produce all the humans that are on the planet.
Mhm, mhm,
And part of that, I think, and correct me if I'm wrong, is that when you see things that are referred to like blighted egg syndrome, where there's like very early
mhm,
miscarriage and things like that,
mhm,
is
mhm,
because the one of the one or a group of the HOX genes have been mutated or are absent even or are cannot support transcription to even get to more than two cells. Okay, and
mhm,
so they're looking at, because the HOX genes control so much, including what Jeff was talking about, microtubule formation for replication. And the information for all of that is contained in these HOX genes. So when you're looking as a geneticist for things that, particularly in genetic diseases, there are certain things that are like totally hands off right now because we know that if we do anything there, we're going to disrupt an enormous part of the whole genetic system. And that's sort of, where a lot of geneticists, particularly, the real developmental folks that are dealing with these issues, the HOX gene is that whole cascade of HOX genes are so incredibly important. Have you been reading much, or do you know much about that phenomenon it's been like 5 years since I read anything on that, but it's incredibly
Yeah.
interesting. I mean, it's like one of the
Yes.
cutting edges on genetics.
hmm yes, we've actually known for quite a long time that a large number of egg sperm unions fail. I think my experience is that it's around 30%. And it's very common. Many couples experience one of these early blighted ovum miscarriages prior to having their first normal child. And it isn't uncommon to have additional miscarriages during, later in life. That is not necessarily at the onset of parenting. Uh, and uh, and I think you're absolutely right that the great variety of these are failures of HOX genes to initiate the early differentiation of the cells that make up the zygote and make up that initial ball of undifferentiated cells that is the first stage in embryonic development. And if there's failure to produce the properly differentiated cells, of which there are hundreds, I mean, it's not a simple matter to to uh, uh, to direct the differentiation process during, initiating the embryo. Uh, if if any one of those groups of undifferentiated cells fail, the 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 whole embryo will abort. And so it's sort of a protective mechanism to prevent the birth of, deformed infants. I think. In any case, it is a fact that it happens. Actually, I did one of my interests was uh, was ultrasonography of pregnancy, of obstetrical ultrasonic demonstration. So I saw hundreds of early embryos with our ultrasonic devices, and we could see the trisomies, for example, which are a clear-cut failure of the chromosome separation process, which again is a very complex process in which the chromosomes uh, are paired and then they're separated and then they migrate within the separating in the cells as they are beginning to separate and form two separate living cells. So in any case, uh, that can a whole series of different aspects of that can fail.
Hey, thanks, Ralph. I appreciate it. Brad?
Thanks. Ralph, thank you for the presentation. I've been tantalized by this stuff ever since I was a freshman in college and read a couple of articles in Science magazine that hinted more was going on in the DNA than people were willing to acknowledge. That was,
Hmm.
in the early 90s when they still called it junk DNA, when they couldn't figure out what it
Exactly.
was.
Yes. Hmm.
My question is, um, you know, conventionally, mainstream evolutionary biologists are very resistant, my understanding, to the idea of suddenlies, these saltation evolutionary events. There was the theory of the hopeful monster, where literally a chicken
Hmm.
lays an egg and out comes something that's not a chicken. It's a speciation event. Today, with the growing understanding of Hox genes, do you think there is a more open-mindedness to suddenlies of many different kinds, including potentially these single speciation events, not things that happen on little islands over a few generations, like punctuated
Hmm.
equilibrium
Hmm.
sometimes talks about, but literally the hopeful monster? Are they saying maybe there's something to this because of Hox genes?
I think you're absolutely right, Brad. I see a great deal of interest being expressed in Hox genes. Now, not much of it is dealing with the philosophical aspects, and as George pointed out, there's the same resistance to discard the established uh, concepts, to throw mud at them, so to speak, uh, because of obviously career protection. I mean, after all, one needs to pay the rent and feed the children. So, in any case, Hox genes, though, is flying in the face of the whole concept of gradualism, and
Mhm.
I think that's the primary origin of the concept of punctuated equilibrium, which, as you suggested, as it was introduced, was very narrowly
Right.
perceived, but that is beginning to erode. And Hox genes have just been around intellectually, as far as we know, for since the 1980s. And so it takes time. Evolution takes time, and evolution is still in progress. And I think I don't think there's any question but what uh, this is gaining significant momentum among geneticists. The paleontologists have been very aware of this problem with the missing links for a long time, but the evolutionists have not been very accepting of their point of view, in my opinion. But I think that is changing.
I do too. I hope so. Thank you.
Okay, I'm going to ask one question from the audience that I'm sort of going to add on a little bit of mine too. John Schreiner asks, are all of the suddenly events specific to the life carrier's plans, including the sequence of the suddenly events? He's asking if the life carriers did it, and my add-on question to that, Ralph, is that when they said they implanted 550 million years ago, they implanted life at three different places on our planet. Are we thinking that that is when they created the original HOX gene? In other words, or
Mhm.
genes that if we eventually get all the way back and begin to
Mhm.
figure out our genetics, will we get back to those original HOX genes, or do you think those developed from something else that the life carriers implanted?
Well, I had that same question, and it occurred to me that rereading from this point of view the paper on the life carriers, I might find something really unique, and I did. There's a section there that clearly states that when the life carriers implant life on a given planet, that they bring with them the entire set of genes capable of producing the entire genome, that it contains the entire genome, capable of producing all of the living organisms that will ever appear on that planet. So all of the necessary genetic information for the future developments of the human race, for example, which will include living to 300 to 500 years in age, which means some fairly significant changes in human biology. The need for all of the other adjustments and alterations in the supporting life on the planet, all of that, from what I'm told in the paper about what the life carriers are capable of doing, is included. So I'm reasonably certain, and I'm very comfortable with what I'm told there, that these genetic capacities, many of which have not yet been expressed on this planet, will lie dormant or lie dormant in the genetic system, in the genome, and they will eventually be expressed.
Okay, that's great. Now that's got a lot of hands up here, but I think David Newfer had his hand up to begin with. So go ahead, David, and then we'll go George and then Jeff.
Well, I've been interested in this not as long as Brad, but in the past couple of weeks, I've been very interested in HOX genes after reading this report. And the thing that interests me was that we know about master physical controllers. These are like mini physical controllers. And there's only 39 of them out of how many chromosomes? Like it's just a few of them. What I wanted to know is, are they all identical, or are our HOX genes identical to those of a like a worm or a bat? So that was kind of my main question. How do they relate to each other?
Yeah.
Are they the same for the 39? Or are they the same? Are they the same for the past millions of years since they've been being used?
As I mentioned in my report, they've been described as being chemically identical in both their chemical makeup, that is their actual anatomical configuration in terms of the exact combination of amino acids and other structures that compose them for some 500 million years, and that's well documented. There's been extensive recent research onto one of the very earliest known living organisms that's still in existence. So so many of them have become extinct over time, but there are a few remaining very, very ancient living organisms, and studies show that there's really no change in the actual gene makeup. Now, there are a lot of changes that have occurred in the genomes. For example, we know that an ancient event was a duplication of the HOX genes that occurred somewhere around 500 million years ago, that gave rise to the 4 different clusters of genes that you saw on those different diagrams. Each of those levels represent a duplicated or incompletely duplicated HOX gene, and in some of the clusters, one or two of the genes are missing. In other clusters, a different gene is missing, but the four of them, the four clusters, do contain a set of identical genes, and the paralogs, which are the vertically oriented location of the genes on the slides that we showed, are nearly identical. Now, there have been a few changes that have occurred with time. For example, the concept of divergence that we referred to, duplication and divergence, those two processes tend to occur simultaneously and actually contiguously with duplications, in that as duplications occur, you have more than one gene that basically provides the same service, the same genetic information in each of the duplicated versions. And that results in a redundancy of genes, which then leads to the diversification in which these redundant genes are sort of free to develop new additional functions, in which probably are mainly involved with the gradualism of evolutionary development. And the gradualism is sort of the other side of the coin of the punctuated changes that occurred. And so Darwin's concept of gradual changes and survival of the fittest is still functioning. That's a genetic process that's well established. But in addition, we have this other genetically driven, punctuating, sudden change capacity that is really very much characteristic of the HOX genes. So once again, back to your question, David, about the number of genes. There are really 13 to 15, it varies slightly, very specific HOX genes that are all very different from one another. And then we have the duplicated sets of genes, such as we have in the vertebrates and mammals specifically, in which there are actually 4 different duplications. As I said, they're not complete. There are some missing genes, and genes can become missing from time to time as a result of various genetic events in the past. For example, I made reference to the rather sudden disruptions that occurred 60 million years ago in the fern genetic genome, and they represent that represents you might consider it a rather turbulent period for the genome, which ended up being quite changed from its original organization. But in any case, it even though we can't document it, there's little reason to doubt that the sudden expansion and growth of the ferns, as they did in a worldwide fashion, was the result of the homeotic genetic system.
Yeah, I just had a question just by you bringing that up. Am I correct that HOX genes are only in animals and not plants? The plants are covered by homeobox. Is that what it's called?
Yeah. Well, the homeobox is a standard feature, so to speak, of HOX genes, in that it consists of 60 amino acids which are essentially identical in every HOX gene. So these are very unique genes that, as we said, seem to have been present on the planet since the beginning of evolutionary life. And
Thank you,
I
Ralph.
use the term evolutionary life in contrast to the recent concept in which some biologists accept the concept that life started 3 billion years ago. Some would even say it's 4 billion years ago. But my understanding is that their evidence is primarily based on metamorphic genetic metamorphic fossilization of single-celled organisms. Now, I think they're reaching pretty far to claim that a single cell could fossilize, pass through metamorphosis of, of rock, which is the result of tremendous pressure and temperature, which produces the very hard rocks that make such wonderful countertops on your kitchen, for example. That I think that's a long reach. But the term evolutionary biology is clearly documented by our fossil record, and it really started with a bang with the Cambrian explosion that happened right around 500 million years ago. Now, there are some disparities in the timing of some of the events, that there's slight variation in some cases with what the Urantia book tells us. But there are many references to 500, 520, 530, etc., million years ago that fit pretty well with what the Urantia book states that it happened 550 million years ago, and that is really the period of time when we have scientific evidence of evolutionary biology occurring.
Okay, we're going to have to move on. I think, Ralph, you meant to say nucleotides and not amino acids, correct? When you were
No,
talking about
well, the nucleotides are part of the amino acids. There's like 3 nucleotides on average per amino acid.
that codes for each amino acid. Right.
Right.
Okay. Right. Okay, George. George is up next, and then Jeff.
Yeah, thanks for this paper. It's so complex. It's ridiculous to think that it's not intelligent. In other words, what you describe is like, evolutionists expecting to find a fully formed typewriter in their backyard, which evolved over the millennia. It's not going to happen. So, but my question specifically was about the possibility. Is there any possibility in this structure you described between homeotic and HOX genes and the whole control mechanism? Is there any room in this mechanism for the intervention of mechanical mind? In other words, the master physical controllers are the source of non-teachable mind, which interacts with living organisms. Did that have anything to do with the sudden appearance or the sudden transformation of phenotypes?
That's a wonderful question, George. And I have no definite insight into that or any kind of scientific, which I say, inklings as to how that might occur. But I think it's one of the mysteries of the interaction between the life carriers and the genomes that they implanted back there 500 million years ago. Now, we're told clearly in the Urantia book that they do not have any permission to intervene or to direct in any direct manner the progress of the genomes, what I think I'm interpreting this correctly, once human life has occurred. It's as though their direct involvement in the control is stopped at that point. But before that, they have considerable latitude in directing animals to migrate, of causing specific, uh, uh, like the lemurs were directed to migrate from North America to the Mesopotamian region because they contained valuable genetic achievement, valuable genetic equipment or genes actually that had very important future would make an important future contribution to humanity, to the eventual, establishment of of organisms with willpower, with the power of will. So, to what extent they were involved in, say, causing these duplications that occurred really early and those that occurred later, such as with ferns, and again with the scorpion. That, in my opinion, is one of the really fascinating examples of sudden evolution in which a scorpion, which was a strictly aquatic organism, is suddenly capable of coming ashore. It has a new lung is grown in its, like, if you look at it from just from a general appearance, it looks like the book lung is sort of down in its chest where our lungs are in reference. I mean, we're very different, of course, from a scorpion, but the overall location of the book lung is almost in the thoracic region. In any case, that was a major achievement. And again, what role the life carriers have in that sort of event, and particularly the duplication events, I think is up in the air. I don't know that we have any clues to that. I don't know if you have any further suggestions on what might be going on.
Okay,
No.
we're going to have to move on to Jeff if we're going to get his question in because we're getting
Okay.
short on time, Ralph.
Well, in
Mhm.
fact, I actually have a suggestion on how that mechanism is, but you'll have to tune into my presentation to get it.
Okay.
Or
Okay.
read my paper, because I do talk about the electrical stimulation, which I think is involved in the gene expression in terms of making sure which genes are expressed. And primarily the HOX genes involve the head, thorax, and abdomen orientation. I'm not sure whether they're in the nuclear DNA or whether in the maternal DNA. 100 copies versus 1,000 copies,
Yeah.
whether that adds to the efficacy of these manipulations. But the preferentially activated genes are either some epigenetic or some electrical environment, because that was I was talking about earlier, and the celestials have ways of intervening in the electrical environment, and certainly the photon to electron environment is another way they can manipulate at the cross energy barriers. So I think it's electrical stimulation to get the right activity. and we do spend some time on that in my presentation, or in the course of my presentation. So looking forward to
Okay. Yeah.
Yeah.
comments.
Well, yeah, we'll absolutely look forward to that. Ralph has reserved the last 3 minutes of this discussion to make a final comment. So,
I'd just like to close with this Urantia book concept and a comment about my own personal experience with it. Preparing this presentation on HOX genes has given me new insights and a greater regard for the versatility of life carriers. They are entrusted with choosing the decimal planets, those suitable planets upon which they are given greater latitude in developing the unique characteristics and improved control over biological molecular activities throughout living organisms as they are doing on our planet. And it was all accomplished here on the planet. We are told that the life carrier core returned to Urantia 550 million years ago, where they organized and initiated the original life patterns of this world and planted them in the hospitable waters of the realm. And in paper 196, entitled The Faith of Jesus, the concluding paper of the book, we are told, be not discouraged. Human evolution is still in progress, and the revelation of God to the world in and through Jesus shall not fail.